Hello, and this is going to be the nervous system. And when you think of the nervous system, here's a picture of a neuron with all the information coming into the soma, and then what's actually happening, and then you have, axons, leaving it and everything like that. Pretty much, these models behind me are what you need to know for this lecture. So because this is lab, all I want you to do is go over what are these different parts. You don't necessarily need to make the connection of how all the parts fit together. What you need to worry about is how do I get all of these structures into my memory, and hopefully at the end of this lecture you'll be able to figure out how do I do that and what's the best way for me to do that. So first, some background information. You have the central nervous system and you have the peripheral nervous system. Central nervous system is going to be the brain and spinal cord. Anything that leaves the spinal cord will be considered peripheral nervous system. And here you can see the central nervous system is in tan, and then starting with these dorsal root ganglia, these little cell bodies that are outside the spinal cord, and then all of these other axons and neurons, those are all going to be the peripheral nervous system. And because this nervous system is very complicated, I do have some background information from your lab class and your lecture class combined into one, just so you can kind of have an idea how do they all fit together. So first we're going to learn about the neuron. Then we're going to learn about the brain. Then we're going to learn about the spinal cord. And then we're going to kind of put this all together in the lab. But for your lecture class, it's a little different. So your central nervous system, you learn about the different parts of the brain, the lobes and stuff like that, the cerebral spinal fluid, what is it, and then how does it actually circulate around the brain. Then you kind of cover what's the spinal cord, what are the descending, ascending tracks, why is one part of it dark, why is some part of it is white, just to kind of, these are all the things that we're going to cover in lecture. We're going to call the names here in the lab. But the peripheral nervous system is very, very, very complicated in lecture and in lab. So let's kind of spend some time with it. You have the sensory division, visceral, and then the somatic. Interesting thing about the sensory, it's pretty much bringing stuff into your body, in towards the central nervous system about feelings, about the outside environment. Motor, and the motor somatic is very, very, very easy. Movement, motor, motion. But the visceral division of the motor division is your fight or flight and rest and digest. And a lot of the times, this part right here is where you run into some issues, because this gets a different name, and we'll go over all these things in a second. But if you noticed, all of these things, they're not in red. So we're not directly testing them in your lab class, but you will have to know them for your lecture. And because the lecture and the labs need to be done hand in hand, it's a good idea to make sure that you cover both of these topics while you're doing lecture or lab. So the sensorant, or afferent. Afferent is another word to say to, because afferent is towards the central nervous system. Efferent, with an E, like exiting, exits the central nervous system. You have the somatic sensory, which is going to carry signals from the skin, muscle, bones, and joints, while the visceral sensory is going to carry signals from the viscera, which is going to be that membrane that surrounds all of the organs in the thoracic and abdominal cavities, like the heart, lungs, stomach, and urinary bladder. And the motor, I told you it's a little more complicated. And the reason why it's a little more complicated is because it's got the somatic motor and the visceral motor. And remember, the somatic motor is just going to be the movement. It's carrying signals to the skeletal muscle. So if you wanted to get up and walk out, or if you wanted to go and walk around, you're going to be using the motor efferent division, and in particular the somatic motor division. And remember, efferent, brain, exit to your muscles via the somatic motor. That visceral motor is also called the autonomic nervous system. The autonomic nervous system is a little strange. And the reason why is because chapter 15 in your textbook, in your lecture class, is just on the autonomic nervous system. So you have an entire chapter just dedicated to the visceral motor division. And the visceral motor division fight or flight, which is the sympathetic, and then rest and digest. And it's kind of important to know what's happening there. So fight or flight, adrenaline, getting the muscles. The blood is going to be shunted to the muscles so you're ready for action. And then the parasympathetic, rest and digest, that blood is going to be shunted from your skeletal muscles to your urinary and digestive systems. There's a lot of systems going on. There's a lot of things happening in the somatic, in the visceral motor division, either fight or flight and rest and digest. It's a very complicated process. This is something we do cover in lecture in embryonic development, and it's kind of important for me to talk about this right now because we are going to talk about the brain, we're going to talk about the nervous system, so it's kind of important to know where does the nervous system come from? How does it develop? And then why is it all these little strings kind of connected? And if you know where they come from, you kind of understand why there's one little part that connects one to another. So by the 4th week in embryonic development, there's a hollow tube. That's called the neural tube. And the neural tube is going to separate from the overlying ectoderm, and then it's going to sink into it. When it sinks into it, it just becomes a tube. And then it grows lateral processes that are later will form the motor nerve fibers, and those processes start to grow out. And then there's a space in the neural tube that becomes fluid, and then the central canal in the spinal cord, ventricles in the brain. This is going to be the beginning of your cerebral spinal fluid and everything that's going to go around the brain. But you think, just by a little tube, this is going to be the beginning structure of all the neurons and nervous tissue. Eventually, you're going to have a neural crest that's going to form from ectodermal cells that lie along the margin of the groove and then separate from the rest, a longitudinal column on each side. Pretty much all this is happening is that these different types of things, these processes are coming out. This is how we're going to give rise to all the different other systems in our body, like the nervous system, skeletal system, skin, and the endocrine system. And that was kind of like a quick look at it. I do have some pictures of the neural tube development, but pretty much by the 4th week, the neural tube is going to show the forebrain, the midbrain, and the hindbrain. When you're doing the brain in the lecture, it's important to know the forebrain is called the proencephalon. Midbrain is a mesencephalon, and the hindbrain is the rhombencephalon. The reason why this is important is because these structures of the forebrain, midbrain, and the hindbrain, these turn into their own unique structures. Different mammals and different types of organisms have different structures within these forebrain, midbrain, and hindbrain structures. And a lot of the times in human development, if you have trouble with the forebrain or embryonic development issues with the forebrain, most of the structures in the forebrain have the same issue. So knowing where everything comes from in the brain and the nervous tissue might not seem like important, but if you go into certain types of industries or certain types of healthcare fields, this might be something you use in a day-to-day capacity. So just a week later, by the 5th week, it's going to divide into 5 different vessels. So now you have 3, and then after another week, they're going to subdivide even more. So the forebrain becomes the telencephalon and the diencephalon. The diencephalon is the optical vesicles and become the retina and the eye. The telencephalon becomes the cerebral hemisphere. So if you've ever wondered why retina and why is vision considered neuroscience, it's considered neuroscience because they both come from the proencephalon. The proencephalon, a week later, divides into the cerebral hemisphere and then the eye. The midbrain is going to remain the mesencephalon, and then the hindbrain the rhombencephalon is going to turn into the metencephalon and the myelencephalon. Myelin, because of myelin, it has a special process of just carrying signals. But we're not going to get into that right now. We're going to start looking at the pictures of the neural tube just to show you how it moves down, and then it starts to make that neural tube, and then you can see how it starts to branch off into individual parts. So after we kind of went over all that, we're finally getting into stuff in red. So first let's look at the parts of a basic neuron, and then we'll kind of understand what's happening. So a soma, it's the control center of the neuron. The soma, cell body. So if you go back to the first part of this semester, when you look at the cell is going to have the cell body. Here, cell body has a special name. It's called the soma. It's got a nucleus, nucleolus, got mitochondria, it has rough ER, has the same thing that was in the cell. It does have some specialized structures. We're not going to get into them right now, but you're just going to know that cell body, you're going to call that the soma. The nucleus in a neuron is very similar. It's going to have the genetic material, but the only problem is it doesn't have centrioles because it doesn't really go through cellular division. What are the ways that information comes to the soma? And those are going to come through the dendrites. The dendrites are what gets signals from outside and brings it to the soma. The soma then can process it and then carry it to the axon hillock. The axon hillock is where all those signals are going to come together. So now let's take a look at that. So the axon hillock, when it's receiving all of that information, it has to reach something called an all or none, or it has to reach threshold. Because if all those signals come in, and if you remember back from your lecture class, some of those signals are positive or making it more positive. Some of them are more negative or inhibitory, and those positive ones are excitatory. That's that balance between what are those signals coming in via the dendrites, coming into the soma. Can it get the axon hillock right to threshold? And once it gets the axon hillock to threshold, then it can send a signal down the axon. The axon can have different collaterals which go off in different directions. So one axon can talk to many different parts of the brain, many different parts of a muscle fiber, and so on and so forth via these collaterals. What makes the axon travel, makes the signal travel so quickly? It's because of the things called myelin. And myelin is going to be made by Schwann cells in the peripheral nervous system. If you remember back in the central nervous system has cells called oligodendrocytes. And the reason why the central nervous system uses those, think about how many neurons are in this little structure right here compared to how far apart they are here. An oligodendrocyte can manage many different types of axons when they're close together in the central nervous system, but when they're far apart in the peripheral nervous system, one Schwann cell, one node. So that is why in the peripheral nervous system and the central nervous system, they have different cells that work with myelin. And it's that myelin that insulates them. And in between those myelins, they have nodes of Ranvier that allows that signal to jump the nodes and travel very, very, very quickly down an axon. And have you ever really wondered how quickly does an axon work? Well, think of it this way. I want to get up. It happens that quickly. Right when you think about something, you can make it happen. If somebody threw something, you would look immediately. That's how quickly these axons are carrying these signals and carrying them to the muscles or glands or organs. So you do have many different types of variations. When you get into neuroscience or you get into more detailed vision or working with neurons, multipolar, have many coming in bipolar, are going to have two different types of poles that look identical. You can't tell which is the dendrite, which one's the axon. Unipolar, which they just have one long strand of dendrites coming in, or anaxonic, which means it just doesn't really look like it has an axon, but it's got many different types of dendrites coming in, and these are going to be in the visual process in the occipital lobe. But pretty much all these different types just tells you that there's many different types of neurons in our body, and all of them have unique functions that helps us take information from outside world, process it, and then have some type of action towards it. So nervous tissue, again, this is just another way to look at it where you see the soma or the neurosoma. You have the dendrites bringing the signal in. You have the axon hillock, and then you have the axon. You do have cells called glial, and the glial cells will be covered in your lecture class. And glial cells are just helper cells. And you're going to learn all about them in AMP1 lecture. This is just a review. You still have the cell bodies, the dendrites, and the axon or nerve fibers. You have the Schwann cell and the Schwann cell nucleus, which is going to make the myelin sheath, and it's just going to kind of go along and keep maintaining the myelin sheath. And then you can see the node, and then the node of Ranvier. One thing I skipped on an earlier slide was this. This is a electron microscope view of synaptic knobs. Synaptic knobs are what one neuron is going to use to talk to another one. It's also what we looked at in muscles, and I said I would cover it later. The synaptic knobs are what talks to muscle fibers. The synaptic knob is going to have synaptic vesicles, which goes across the synaptic cleft, which allows it to talk to a target organ. Now, let's look at what are the models you're going to be tested on. So first, you have this one. This model right here is the same one that you see on the slide in front of you. And the main thing, and the thing you can't really see here, is that right there is a synaptic knob. That's a dendrite. But here, I can show you in a little bit more detail. This right here, you can see it is rubbery. If I go right here, that right there is hard plastic. So all the ones that are rubbery are going to be your synaptic knobs. And if you're like, is that true for every single model? Yep. These that are rubbery, that move and do all this other stuff, those are synaptic knobs. The hard plastic is going to be your dendrite. So it's an easy way to kind of see and feel and look and say dendrite, 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 synaptic knob, synaptic knob. All of that's going to be your soma. You have your nucleus with the nucleolus. Your signals coming down here to the axon hillock, then the axon. You have your myelin sheath and node of Ranvier. On this one, you still have your synaptic knobs and dendrites. Nucleus is down there. The axon hillocks there. Here you can see a cross section of your axon. You can see the Schwann cell and the Schwann cell nucleus, and then the myelin sheath. This is another view of them. You don't need to memorize these, but you do see they have rough ER and smooth ER. They do have Golgi apparatuses and complex and vesicles. They have lysosomes, peroxisomes, and different types of concentrations, and they do have mitochondria. You don't need to know it for the for the final, but you just need to know that a cell has certain types of structures, just like a neuron has certain types of structures as a normal cell in the body. And this one is just this view right here, where it has all the different things. You can see the node of Ranvier, you can see the Schwann cell, you can see the myelin sheath, and you can clearly see the axon running all the way down the cell. And then this is just a close-up view of the neuromuscular junction. These are the muscle cells. And then here's your Schwann cell and then your myelin sheath. You do have your synaptic knob. Your synaptic knob, I mentioned, has synaptic vesicles. And because it's neuron to muscle, the neurotransmitter here is going to be acetylcholine. That's that ACH. It travels past the synaptic cleft. There's receptors for acetylcholine that then open up ion channels and allows the ions to move down, and then they move to the T-tubules, depolarizes the terminal cisternae, and allows calcium out of the sarcoplasmic reticulum. And this is just a view right here of what the neuromuscular junction looks like. You can see those striped or those striations of a muscle fiber. And this just explains what's happening where it comes down. It allows the synaptic vesicles to fuse to the synaptic cleft. It travels past the membrane, goes through the synaptic cleft, goes onto the acetylcholine receptors, opens up, causes depolarization, and that depolarization opens sodium and potassium channels that allows that signal to propagate down the cell. And eventually it makes it all the way into the muscle fibers, and then that's pretty much inside a neuromuscular junction. And we do have and we do have a model that has the synaptic knob, but it's only on the Lake Worth campus, so normally I don't have it, but if you do see that little synaptic knob, the only thing you need to know is synaptic knob, synaptic vesicles, and that is the end of the neuron and everything you need to know for your final. That includes the neuron. And if you look, these are the two models we're going to have on the neuron. So please study the YouTube videos, make sure that you study the pictures, have somebody point them out to you, and do your best. And as always, good luck.